All right, well, welcome to microbiology. This is our first microbiology lecture, and we are going to start right here at chapter 1. So, kind of at the beginning of chapter 1, it's an introduction into what is microbiology and a little bit of the background of microbiology. So, at the very beginning, what we're talking about is kind of what we're going to be talking about throughout this entire semester, throughout the entire class, is we are going to be talking about microorganisms. Sometimes they're also called microbes. So microorganisms, if we kind of split that word apart, we have micro meaning small, and then organisms. So small organisms are small kind of living things. And it doesn't necessarily mean living things specifically, because we're going to take a look as we move along that we are also including viruses when we study microbiology or study microorganisms. And technically, viruses are not living things, and we'll get into that into more detail, but it's viruses and lots of very small living things. So most microorganisms that we're going to talk about are very, very small organisms. Usually they're too small to be seen with the unaided eye, so too small to be seen. How about without a microscope? So too small to be seen without a microscope. Some can be seen with a microscope, so we have some, molds and things like that that are large enough to be seen without a microscope. But mostly what we're going to be talking about are things that need to be using a microscope for us to kind of see and to investigate with. So we'll be talking about microorganisms, so that's everything from bacteria. We'll be talking about viruses, as I mentioned, so bacteria. We'll be talking about viruses. We'll be talking about fungi. Molds. We'll be talking about protists. We'll be talking about helminths. So rather than listing them all here, we're going to be talking about basically things that are very, very small or have parts of their life cycle that are very, very small. So for example, helminths, those are what we kind of think of parasitic worms, for example, which the worms can be seen without a microscope, but the other parts of their life cycle oftentimes cannot be seen without a microscope. So we're going to be looking at all kind of really tiny living things. So at the beginning of the chapter here, we talk about how, as we're working through this semester and talking about microbiology, we're going to be talking about all of these different little things, but we want to take a look at them that many of them are not even harmless. So oftentimes when we think of microorganisms or microbiology, we think of kind of bad things that are going to make us sick or make other things sick, like animals and things like that, which is true. We do have many viruses or many bacteria that can cause illness. However, most of these microorganisms that we're going to be talking about are harmless to humans and animals, and a lot of them are actually helpful. So we can see going back hundreds and hundreds of years, basically to recorded time, as far as we know it, where microorganisms are being utilized for fermentation. So for example, for making different fermented beverages, making cheese, making beer, making wine type things, making yogurt, kombucha, ginger beer, things like that are all being made very, very long time ago, utilizing these microorganisms in beneficial ways, and actually in ways that we today try to get into our systems, like for example with fermented vegetables and things like that. So if we didn't have microorganisms, we obviously don't want to wipe out microorganisms. Oftentimes when we think of microorganisms, we think about the illness, bacteria and viruses, and we say, oh, we want to get rid of these. But it's important that we study them, and that's what microbiology is. It's the study of these microorganisms, and that's because many of them are helpful, or at least not harmful. And so it's important that we understand them, and then we can utilize them to benefit ourselves or benefit the plant world. And we have various ways that we use these that are beneficial. So for example, we utilize them in fermentation. And I already mentioned some examples of uses of fermentation. This is where we take our carbohydrates, and we're going to take those carbohydrates and allow these microorganisms to ferment them and turn them into different gases and organic acids. And then that's beneficial to us. So we put yeast in with flour and some other things, and then we get bread at the end after we cook it. So that is helpful to us. And so we see that even a very, very long time ago, people used these microorganisms. They didn't realize what they were doing. They didn't know that they were, little tiny organisms that are being used to make their fermented food or their fermented beverage. But that's what they were doing. Even if they didn't know that they were there, they were still doing it. And so we can see some examples of that when we talk about, as your text does, when it mentions Otzi the Iceman. So if we take a look at Otzi the Iceman, what we're talking about is this mummy that was found frozen in the Alps, in the Australian-Italian border. So when they found Otzi, what they discovered is that he was infected with a parasite and also perhaps had Lyme disease. So he had a parasite, and perhaps he had Lyme disease. And what he also had with him was that he had some fungus, that if you use that kind of the woody fruit of this fungus, then it can help to have antibiotic properties. So he perhaps used the fungus and used those antibiotic properties of the fungus in order to treat himself or to help. It also showed that he was burning herbs in his skin, cutting his skin, and then filling them with herbs and burning those herbs to try to help, likely to help with his health as well. So even back then, during this period of time, we see the use of microorganisms. So what we are going to do is we are going to talk about kind of the background, so kind of what they thought about microorganisms a very long time ago before we really could see them with a microscope. So when we're talking about these things, again, a very, very long time ago, people didn't know that they were microorganisms, that they were little tiny individual single-celled organisms or multicellular organisms that were doing these things that they were doing. So before they knew that these things existed, they were still somehow talking about them. And so what we were talking about, for example, they could see that there was something to do with this spreading of illness or the spreading of disease, or that they could ferment certain things. And so ancient civilizations actually understood that diseases could be spread. So even back to ancient civilizations, they knew or understood, maybe they understood, disease could be spread. And so this wasn't proved, of course, until the invention of the microscope, so where they could actually see them. But we knew that they understood this because they did things like quarantining. And so they would take people that were diseased and they would put them all in the same area. Unfortunately, they probably had already spread the disease because by the time they were showing symptoms, they probably have already spread it to other people. However, they did quarantine people, but at that time they were speaking of it in different terms. So we see that the ancient Greeks, as well as the ancient Romans, spoke about what we now call microorganisms in a way of miasmic odors. So miasmic or miasmatic odors. And so when we're talking about miasmatic odors or miasma hypothesis, what they are really talking about is this kind of bad air. They just thought that there was kind of a bad air surrounding somebody when they were ill. Or they would think that perhaps the gods or the god would be angry. And so then there would be this kind of bad air surrounding the town or the area. And so then in Rome, they built these aqueducts, or they built sewers to kind of get rid of the bad air of people using the restroom, for example. So they created these aqueducts or these sewers to take the waste away from the city because they saw that there was something to do with the waste creating this kind of bad air as they saw it, and then causing illness or causing disease. But of course, they didn't know that it came down to these little tiny organisms. So we didn't really get to this idea of actual organisms or something that's not supernatural until we get to Hippocrates. So Hippocrates, around 460 to 370 BC, he really is he's considered the father of Western medicine. And he's considered the father of Western medicine because he was kind of the first person to say that disease is not supernatural. So disease is not supernatural, meaning that some god is not angry and then striking you down with illness. He's saying that it's not this supernatural thing, that disease really comes from other people and the environment. So that disease comes from other people and the environment. And so he's saying, okay, it's not this kind of God principle, but instead they're saying like he could see that it's coming from other people, people coming into the town or people leaving the town, and the environment around them, things that they are around. So Hippocrates is credited for the Hippocratic Oath as well. So the Hippocratic Oath, which is the oath that all new physicians pledge. When they become a doctor, they pledge the Hippocratic Oath, which is basically saying that they are promising to treat patients and not cause harm. So first, cause no harm. And he also wrote the oldest books on medicine. So Hippocrates, again, the father of Western medicine, kind of the first physician-minded type of person. And so he did write some of the oldest books. How about wrote? The oldest books on medicine. So then again, Hippocrates kind of moved us from this miasma idea and the gods being angry kind of idea into actual Western medicine. After Hippocrates, then we had Thucydides. And Thucydides was similar in time frame here. We're talking about the 460 to 395 BC. He was Greek. And he's considered the father of scientific history. So the father of scientific history. And so the reason that he's considered the father of scientific history is because he advocated for evidence-based approach. He saw that there was a cause and effect. So again, rather than this kind of miasma idea where there's just kind of this angry cloud of disease that somebody can walk into or the gods have put on someone, he said, let's take a look at this. Hippocrates had already said that there's this connection between other people being ill and the environment, what people are around. And then Thucydides said, look, there's actually a cause and effect here. So we'll say he advocated for an evidence-based approach. So he saw and talked about cause and effect. And that you could analyze this, that you could see that when one person was sick, if another person came around that person, then they then got sick. So that's the cause and the effect. They were near the person, and then the effect is that they get sick. Or they were near something in the environment, maybe an animal that was ill, and then they got sick after that. He did also notice that survivors of a particular illness did not get sick again. So he was the first person to note immunity. And this is because he himself became immune. So he was he lived during the time of the Athenian plague. So let me write that down here. So the Athenian plague. And the Athenian plague killed about 1/3 of the population in Athens. And so he actually got the plague, and he was cured of the plague. He became healthy again. And then he noticed that when he went around others that currently had the plague, that he did not get sick again. So he was able to help take care of sick people because he didn't get sick again. And so these were his first kind of cause and effect. He noticed that that people didn't get sick again. So he noticed that survivors didn't get sick again. So again, he didn't quite understand why this was, but he did make that observation. And so that was kind of added to the information that we now have on microbiology as a whole. So then after Thucydides, then we have Marcus Terentius Varro. So what Marcus Terentius Varro. So he was around about 116 to 27 BC, and he was a Roman writer. And so what Varro did, he was one of the first people to propose that there were things that we could not see that then would cause disease. So he suggested that things we cannot see can cause disease. So he was kind of taking what not only Hippocrates but Thucydides had kind of brought forward, was that it wasn't just kind of this bad air idea, but he said even further that he believed, or he kind of proposed the idea or suggested that there was something in the air, that there was actually something in the air of the people that were ill or the environment that then would cause the disease. And then further, Thucydides is going to assist in saying that once that person caught that thing, and then they went through the process of being ill, that they didn't get ill again. And so then Varro then is suggesting that we could identify these little things. And then he called them animalia minutia. So animalia minutia. Or very small animals or minute creatures. So he was saying that there's something that is in the air. We can't see it. It's too small for us to see, but it is the thing that is passing disease from one person to another or from the environment to the other. But at this point, we still can't prove this. It's not until we get to Antony van Leeuwenhoek. Antony van Leeuwenhoek, who is around about 1632 to 1723. And what Antony van Leeuwenhoek did, he was Austrian. He is the first one to develop a lens that was powerful enough to see these microorganisms. So he wasn't even really technically a scientist. He was a cloth merchant. And he developed a lens. So he developed the first lens that was powerful enough to see microorganisms. So he was actually the first one to develop the lens itself. So he then is credited for creating the first microscope. So credited creating first microscope. Because they don't really know who kind of put all of the pieces together, but he is the first one that came up with a lens that allowed people to see these microorganisms. And then what he did was he took a look at a drop of rainwater, and he saw things floating around in it. We know now that was bacteria and protists, but that back then he saw all these little single-celled organisms. So he looked in a drop of rainwater, and he saw, all right, hang on, I can erase that. Single-celled organisms. We now know that he saw bacteria and protists. And he called these animalcules, or wee little beasties. Sorry there. So that's what he named them.
So he was really the first one to really prove what Varro, Terentius Varro, had said, where he said that there are these little things that we can't see that are causing the disease. And what he did then was he kind of put a visual to those little things. He confirmed that, yes, there are these little things that we can't see. Now we can see them because now I've created this lens, and this lens makes them large enough for us to be able to see. So he did drawings of these little organisms, and that's how we know now that they're bacteria and protists. We can kind of take a look at the drawings, compare them to things that we know now, and then his contribution is then going to really open things up as far as microbiology is concerned, because then people are really able to take a look at these organisms and study these organisms. However, it does take about 200 years before we get into what's considered the golden age. So the golden age of microbiology. So the golden age of microbiology is about 1857 to 1914. And when we're in 1857 to 1914, this is where we get to two very, very famous microbiologists. Those two famous microbiologists are Louis Pasteur and Robert Koch. So you probably have heard of at least Louis Pasteur. And Pasteur, he was French. He was a French scientist. Wow, okay. Here we go. So he was a French scientist, and he really advanced our understanding of microorganisms. So he was really the one that proposed that individual strains had unique properties. So he would talk about individual microbial strains. He was able to identify that there were different little tiny organisms out there that we couldn't see without the use of this lens, without the use of a microscope, and then that each of them had their own unique properties. And then he also demonstrated that fermentation is due to microbes or microorganisms. So he showed that individual strains of microbes, we'll shorten it up, had unique properties. He also showed that fermentation was caused by microbes. So he was able to isolate the microbes from the fermenting products, isolate them, take a look at them under the microscope and see them, and then he was able to grow more of them and show that they had different or unique properties. He, of course, then also invented pasteurization. Hence the name pasteurization. And pasteurization is this process that kills microbes that cause food spoilage. So this, of course, is important because we want to be able to get rid of microbes that are going to cause spoilage in food and then cause disease. So he invented pasteurization. We're going to talk a lot more about Louis Pasteur and what he figured out in microbiology for us. And we're going to talk a lot more about pasteurization in further chapters. Lastly, he developed the first vaccines for treatment. So he developed vaccines for treatment as well. And we are going to talk a lot more about vaccines. We're going to talk about the specific vaccines that Pasteur also came up with. So just a little bit now that you should know about Louis Pasteur is that he did connect individual strains of microbes, took a look at individual microbes, and then saw that they had unique properties that were separate from others. Showed that fermentation was caused by microbes. He invented pasteurization to kill these microbes, the ones that are causing food spoilage, so that people could have food that lasted longer. And then he developed vaccines for treatment. So he worked on noticing that they had unique properties and then using those unique properties to get rid of them. And then secondly, during this golden age of microbiology, we had Robert Koch. So Louis Pasteur and then Robert Koch. Robert Koch was a German. And he showed that there's a connection between a single microbe and known human disease. So a connection between a single microbe and a known human disease. So what Robert Koch did, so we already had Louis Pasteur. Louis Pasteur is showing that individual strains had different properties. Then Robert Koch took that a little bit further, and he said that there are these single strains that have these unique properties, and then we can show how that connects to our known human disease. He could say this is the individual microbe, and it causes these symptoms, these things that are in a human that we call this disease. So, for example, he discovered the bacteria that cause anthrax, which is Bacillus anthracis, the bacteria that causes cholera, Vibrio cholera. So he was able to connect a specific microorganism to a specific disease. And so this was important because we had Louis Pasteur here that's isolating these different things, and then Robert Koch, and we're going to talk a lot more about Robert Koch, and he had different postulates where he was able to isolate these different things and then connect them to a specific disease. So that then what you could do is you could see a person that is diseased, try to isolate the bacteria from them, and then you could see that connection between that particular bacteria, that particular disease, and now we've kind of come full circle in the fact that we know that we have these little tiny things that we can't see without a microscope. We know that they have different properties from each other. We know that they can ferment things. We also know that they cause disease, and we can actually isolate them from diseased individuals. So all of these people really contributed to our understanding of microbiology. And moving forward, what we're doing is we're kind of taking what they had kind of laid for us and going further and further into understanding these different microorganisms in even more detail as we move forward. So everything from the protists to the bacteria to the viruses, all the different types of organisms that we can't see with our unaided eye without a microscope, and really kind of isolating them and taking a look at their characteristics, now not only from a human disease perspective, but also from a genetic perspective. And we'll take a look at all of that throughout the semester. So this was kind of our introduction to microbiology, an introduction to the different ancestors of microbiology. And we're going to move on to our systematic approach in the second part of the chapter.